Hello friends, how are you? Today I am going to present second part of my lecture on that is the human vision. If you remember, last time we have studied that is about the structure of the eye, and the last point which we discussed about that is the difference between the rods and cones. If you look at this slide, you will find that we have studied this so many properties about rods and cones. Now we are now going to study here about color vision. There are different theories of color vision. Very commonest and which is most accepted theory is Young-Helmholtz theory of color vision. According to this theory, it is accepted that there are three kinds of cone photopigments and are sensitive to one of the three primary colors. Now, which are these three primary colors that all of you know? That is blue, green, and red. The blue cone system contains S pigment. S pigment means these pigments are sensitive to short wavelength of the light. Contains pigment, that is blue sensation pigment, which responds to short wavelength about say 440 nanometer, senses the blue color. The green cone color contains M pigment, means green sensation pigment, which correspond to middle wavelength about say 535 nanometer and senses the green color and less to yellow and absorb light at green portion. Third type of the say cone which is meant for perception of red color is red cone system, which contains here L pigment where red sensation pigment, which responds to large wavelength at or more than 535 nanometer, so senses the red and yellow color and absorb light at red portion. Now, what is the photochemistry of the vision? The light sensitive photochemical is found in the outer segment of what that is the rod and cone. Rod contains rhodopsin and cone contains three color pigments, that is the red, blue, green, that is the color pigment are sensitive opsins, what we call as the iodopsin. Function of cone pigment is the same as rhodopsin except for spectral sensitivity. Both rhodopsin and color pigments are conjugated proteins. These are present in the membranes of the disc as transmembrane protein, and there is high concentration of this pigment, about say 40% of entire mass of the outer segment. The photosensitive compounds in the eye are made up of protein opsin and retinine. That is, this type is retinine 1 in humans and retinine in some of the animal species. The retinines are aldehydes of vitamin A1, are called as retinals. The A vitamins themselves are also alcohols, therefore called retinols. Opsins of rhodopsin or visual purple is called scotopsin. Opsin of the cone is called photopsin. Rhodopsin is maximally stimulated at wavelength of 505 nanometer. The blue, green, red sensitive cone pigments are maximally stimulated at wavelengths of, just we have seen, about 445, 535 and 570 nanometers. And rods are stimulated at say approximately at 507 nanometers of light wavelength. Now, just I have told you that is they are the Thomas Young and YOUNG-HELMHOLTZ say theory, which they postulated that according to this theory, the color vision in human is responsible due to the three kinds of cones, each containing different photopigment and maximally sensitive to one of the three primary colors. Three types of cones in humans we have seen, mean for red cones. They are also called as the erythro with a 570 nanometer sensitivity. Green cones, they are called chlorolab, that is sensitivity 535 nanometer, and similarly that of the blue cone, they are called the cyanolab with say light sensitivity to 430 nanometers. What is the photochemistry of vision? Light sensitive chemical in the rods is called rhodopsin and in the cones called the cone pigment, which have slight different composition than rhodopsin. Both chemicals decompose on exposure to light and excite the nerve fiber from the eyes for perception of the image formed on retina. The principles of photochemistry of rhodopsin can be applied to the cone pigments. The outer segment of the rod, as which is mentioned, say that projects into the pigment layer of the retina has a 40% concentration of light sensitive pigment. We call it as rhodopsin or visual purple. This substance is a combination of the proteins cotopsin and the carotenoid pigment retinol, for example, that is the retinine. Two forms of, say, retinal can occur as 11 cis retinal and 11 trans retinal. Retinal is 11 cis retinal type. This form is important. This form of retinal is important because only this form can bind with scotopsin to synthesize rhodopsin. When light energy is absorbed by the rhodopsin, it is immediately decomposes within a fraction of a second, as shown in the figure, from, say, cis form of retinol to all trans form. All trans form separates from protein scotopsin since it does not combine with reactive sites on the scotopsin and hence immediately form beta-rhodopsin. Beta-rhodopsin is extremely unstable and decays in nanosecond to lumi-rhodopsin. Within microsecond, lumi-rhodopsin decays to meta-rhodopsin. Now just give a look to this figure. It shows here rhodopsin-retinal visual cycle. Just give a look to this figure, which gives the idea about the rhodopsin-retinal visual cycle, or what we call it is the bleaching of, that is the pigments in cone and rods. Now as just we have seen, the light energy is absorbed by the rhodopsin. Then we have seen it is converted, say it's a 11 cis transform is converted to, that is 11 all form, and say now it cannot combine here with, say, remain combined with the scotopsin, and it gets separated, and further, as I have just mentioned, it is, say, converted to the beta rhodopsin, then it is converted to the lumi-rhodopsin. Further, it is converted to the meta-rhodopsin, and that is there are two types of the meta-rhodopsin: meta-rhodopsin 1, meta-rhodopsin 2, and as we have just mentioned that is, say, a separate opsin is formed, or and here say that all trans retinal form, say, is converted to the 11 cis retinal form, and it combines with opsin once again to form rhodopsin. Similarly, there is a pathway. This all trans there is other pathway, that is all trans retinal is formed to all trans retinol, which is vitamin A, which is converted to the 11 cis retinol. In both reactions, we require here, that is the enzyme isomerase, and that, say, form 11 cis retinol is combined with opsin to form rhodopsin. Next is that within milliseconds, that is a meta-rhodopsin 1 decays to meta-rhodopsin 2. That is what we call it as a activated rhodopsin, and it is, which is important because it causes, say, that is the stimulation of the, say, optic nerve to perceive the image. Finally, within seconds, meta-rhodopsin 2 completely splits into scotopsin and all trans retinol. Now, how this, say, rhodopsin is formed? How, in what way the reformation of rhodopsin takes place? This process requires metabolic energy to convert all, say, trans retinal, catalyzed by enzyme retinal isomerase. Now this formed 11 cis retinol automatically combines with scotopsin to reform rhodopsin, which remains stable until it is decomposed by absorption of light energy. Now once again, please remember here, this rhodopsin is, say, that is stored in, that is, say, the outer segment of, that is the rods. Then what is the role of vitamin A? The vitamin A is very important in the formation of rhodopsin. all trans-retinal can be converted to 11-cis retinol form by second chemical route as shown by enzyme retinal isomerase. First, all trans-retinal form is converted in all trans-retinol, which is one form of vitamin A. Just try to understand. First, what is the role of, say, vitamin A in formation of rhodopsin? Now, all trans-retinal can be converted to 11-cis retinal form by second chemical route by the same enzyme retinal isomerase. First, all trans-retinal form is converted in all trans-retinol, which is one of the form of vitamin A. Next, all trans-retinol is converted to 11-cis retinol by that is enzyme retinal isomerase. Finally, this form 11-cis retinol is converted to 11-cis trans retinol, which combines further with scotopsin to form new rhodopsin. Now here is one important thing just we must remember here, that vitamin A is very important in formation of that is the rhodopsin. However, if suppose that there occurs say severe deficiency of vitamin A, which causes here night blindness, or deficiency of vitamin A is the major cause for that is the night that is the blindness. Mainly remember that night blindness is say that is very common in the males as compared to their female counterpart. Vitamin A is present both in the cytoplasm of the rods and in the pigment layer of the retina. Therefore, vitamin A is normally always present to form new retinol when needed. Conversely, when there is excess retinol in the retina, it is converted back into vitamin A, thus reducing the amount of light sensitive pigment in the retina. This is the importance of say, vitamin A as in formation of rhodopsin. Now you can see in this figure that is the color sensitivity to say that is the to different cones. Now it shows here say integration of formation of red, green and blue cones. Now it shows say that is the stimulation of that is the respective cone with say as shown here with the different wavelengths. Just I have mentioned about here color blindness, which is mainly caused due to the deficiency of vitamin A. I have just mentioned here about the color blindness, which is caused due to the deficiency of that is vitamin A. Let us classify now this say that is the color blindness. Now it is classified in the three categories: anomalous trichromatism, its occurrence is 5 to 6%. There are say three subdivisions: protanomaly, which indicates that is a red color weakness; deuteranomaly, which indicates here green color weakness; and titanomaly, which indicates that is a weakness for blue color. Second type of color blindness is dichromatism. Its occurrence is about approximately 2.6%. There is say the first subcategory protanopia, means it is a totally say that is the red color blindness; deuteranopia, which indicates here that is the blindness for green color; and titanopia, it is the blue color blindness. The last category is monochromatism, which is extremely rare and its occurrence is approximately 0.003%. And this say abnormality indicates that complete color blindness and cannot perceive any type of color except black and that is white. Now there are different tests for detection of that is color blindness, which are very common, which are used: Holmgren's wool test, the Edridge green lantern test, and Ishihara chart. These are very easy to say that is the test, that is the color blindness. Now I will show you that is the Ishihara charts. Now here say you will find the say certain color plates with say different say color combination in the form of dots, and you can see here that is certain numbers which are formed with the different color combination. Individual has to identify this color. This say that is the say figures. If he is able to identify this figure, then it indicates that say that is the say person can differentiate various say types of color. For example, in the first plate you can see the figure 7, next here 13, next 16, then next 18, middle one 12, and last is that is the 9. Means if you are able to read this all plates and these numbers, then it indicates that you are having normal say that is the color vision. Now regarding say that is the congenital dichromatism, just I have told you that is a type that means cones themselves are normal, but one of the them contains that is the wrong pigment. This we have seen its type deuteranopes, protanopes, and that is the titanopes. Then say after that, now what is important part remaining is that is image forming mechanism which is present in the eye. Now let us concentrate here. Just once again we will go back to the basic structure of the eye, as we have seen that it is say eyeball is say divided in two say segments, posterior segment and the anterior segment. Now this anterior segment, what does it contain? It contains say cornea, then say that is the lens, and it is followed by posterior segment. This say anterior segment is very important in formation of the image on that is the retinal surface. Then some of the important parts which are needed for discussion is that is the innermost say aspect of the eyeball in the posterior segment is the retina, and say on the retina you will find at the periphery say the maximum numbers of rods are present, and that is say below say that is the optic disc where you will find that there is a maximum accumulation of that is the say cones, and this part is called as the macula luata. Now I also told you that is the eye is divided into compartments, that is what are two components, that is the optical division and that is a neural division. Now this part we will say it is the say optical division, and this part where you will find that there is a entrance or you may say exit for central retinal artery, veins and nerves. This part is very important which conducts the information about say that is the image which is formed on the retina towards brain for perception of this image. Now how say that is the image is formed? Image is formed by the simple manner. The light strikes this part, what we call is the cornea, passes to the say that is the anterior chamber, posterior chamber, passes to the lens and forms the image on the say retinal surface. Coming to here say, now while passing the light from one medium to other medium, we know that there occurs say reflection or say refraction. Now since when light is passing through say the transparent medium, there occurs the refraction of the light as shown in the figure. That is say you will find that the light say when it is passing, it gets refracted through the eye. Now it follows this pathway, that is the bulbar conjunctiva, cornea, aqueous humor, lens, vitreous humor, ganglionic cell layer, inner synaptic layer, bipolar layer, outer synaptic layer and photoreceptor layer. I mean, in short that whenever the light say passes from air to the say that is cornea, it gets say that is the refracted. I mean, in this way, this figure shows here the light which is passing. It follows say this pathway in the eye. Coming to how this image is formed and how it is interpreted in the brain. Now suppose that we are looking at this object 1. Okay, the light rays passes from corneal surface, then to say through that is the say lens. This is the, as we have seen, this is the say biconvex lens, and here the image is formed on the retina. Now remember, this image which is formed on the retina, it is say exactly opposite mirror image of this. In other words, I would say this image is formed in the say that is the in the head in the downward position, and here how it is say that is interpreted, that is interpreted as that is head is the in the upright position. How this image is formed and it is interpreted in the brain. Now when we observe any object, for example, the object shown as number 1, say, and its image is formed on the retinal surface, that is where we call this the head down position as compared to the, say, that is the object 1, and say the image which is formed on the retina, from that here say photoreceptors are stimulated and their impulses are conducted from optic nerve and they terminate to their, say, the target say portion in the brain, what we call is the occipital cortex, and now the image is perceived at, that is a head upright position, means say that is what we have, say, that is observed, that is say observed as it is, and by the observed as it is in the brain. Now one important thing, now we are not going to consider here say all details of the optics, but we know that we will consider here, say, that is two lenses. Now at my left side you can see that is the, say, biconvex lens, and at the right side you can see here that is the biconcave lens. Now as we know that when light rays passes from say biconvex lens, it, they are focused at one point, and in the biconvex lens, say, they are focused at the opposite side. Mean what is the property of, say, that is the biconvex lens is to focus the rays at one point, and that is the property of biconcave lens is to cause a diversion of, say, that is the light rays. Now the, say, the, as we have learned, that is every, say, that is the optical apparatus or the lens is having a center, and say, for example, now these lenses are having center at their, say, at vertical, say, optical axis, and say the image is formed or the rays are converging at this point from F, and now suppose that this is, say, our point, that is the, say, optic center. This, say, distance from optic center to the focus point we call it the focal length. The focal length, it is nothing but it is a distance from the optical center of a lens or a curved mirror to point where light rays from a very distant object, say, converge. Now, similarly, this is a convex lens, you can see. Now the focal length in this case is, that is about 20 centimeter, and that for, that is, say, biconcave here, that is lens here, the focal length is 20 centimeter. Now we can express the, say, the power of the lens in terms of, say, diopters, that is capital D stand for diopter. Now how we determine? It is nothing but it is a reciprocal of the focal length. Now we can see here, now this, say, the focal length we have converted into, say, the units of meter. If you take, say, that is a focal reciprocal of this focal length, suppose that now it comes as 1 divided by 0.2 meters, means the power of the lens becomes 5 diopters. Similarly, in this case also, say, that is the power of the lens is, that is the 5 diopters. Now one important point, eye is having, say, different, say, that is the, say, that is the refractive medium. For example, cornea, vitreous lens, and they have, that is the different, say, that is the, say, what we call it as, that is the, say, dioptric powers. For example, in human eye there are several structures, for example cornea, crystalline lens, etc., which act as lens. That is, human eye has a compound lens system. Now we come across, say, one term, that is the reduced eye. What is this reduced eye? As we know that in human eye there are several structures, for example cornea, crystalline lens, etc., which acts as lens. Just to mention here, that is, human eye has a compound lens system. Center of all these lenses, that is the center of optical system, is called nodal point of the eye, which is, say, 17 millimeter and lies in front of the retina. What does it mean? Image is focused on the retina Hence, the focal length is about 17 mm. And the power of the optical system of the normal eye is how much? Means we have to consider the 17 mm into the, say, that is a meter. Okay, means, and similarly we say determine that is the power of the lens is nothing but simply it is a reciprocal of focal length, means 1 divided by 17 divided by 1000, means that is the, say, that is the dioptric power of, say, the total, say, that is the lens system of the eye is, we consider is as, that is the 15 diopters or 16 diopters approximately. Of these 59 diopters, cornea contributes 43 diopters and lens, say, accounts for 16 diopters when removed from the body. In vivo, the lens accounts only for 10 diopters. If the refractive surface of the eye are algebraically, say, added together and then considered to one single lens, the optics of the normal eye may be simplified and represented schematically as reduced eye. In the reduced eye or schematic eye, it is considered that eye has a single spherical surface, that is the lens, of radius 5.6 mm, separating two media of refractive indices, that is 1 for air and 1.33, that is for aqueous humor, and that is situated at 1.4 mm behind the cornea in aqueous humor. The nodal point of this eye is situated at 7 mm behind the anterior surface of the cornea. This figure shows here reduced or schematic eye, in which, say, this is an eye with simplified optics. In this eye, the overall refractive media or power during rest is represented by single convex lens placed at 15 to 17 mm in front of the retina with total refractive power of 59 diopters. The image formed on the retina by refractive media is true small inverted image which is corrected in position by cerebral cortex as, that is the, say, head in the upright position. Now coming to the one important aspect that is we come across all the time. What is this blind spot? The blind spot is what? That is also called as optic disc, where the optic nerves converge and exit the eye. No light sensitive cells to detect light rays, results in a break in the visual field and known as blind spot. What is the peculiarity is that this portion as shown here in this figure, say, that is exit or entrance point of the nerve and the blood vessels, which what we call is the optic, say, that a disc or blind spot, which is totally devoid of the photoreceptors, and never a image is formed at this area, and hence this is called as a blind spot. Now, which are the, say, abnormalities of the eye? Now, these are the very commonest type of the abnormalities that we know. That is the myopia, hypermetropia, presbyopia, astigmatism, and these are the color blindness which I have just mentioned. Now, when the, say, these, say, four, say, abnormalities, say, occur in the eye due to the disturbances in the optical system of the eye, then we call it as a refractive errors. Now, what happens in the myopia is nothing but, or we call it as, that is a nearsightedness. Second is the hyperopia, or that is we call it the hypermetropia. It is a farsightedness. And third is the presbyopia. This is the age-related failure of the lens to accommodate, or this is inability of the accommodation. Accommodation is nothing but inability to adjust the focal length of the eyeball. Astigmatism is nothing but it is the, say, distorted vision due to the irregular shape of lens or cornea. Means what you will find that the corneal surface and the lens surface become irregular in different meridians, and the image which is formed on the retinal surface is perceived as a blurred image. Now this is the, say, normal, say, eye with a normal vision. We call this as a emmetropic eye, and whenever these, say, refractive errors are produced, then such, say, eyes are called as a amytropic eye. Now this is in the nearer, say near sighted, say that is the eye. That is what you will find is that as compared to the normal eye, there is the increase in the say anterior posterior axis of the eye, or in short there occurs the elongation of the eyeball. And how it is corrected with the say lens, that for that we use a concave lens. Then this is the far-sighted say eye or hypermetropic eye. Say in uncorrected say that is the eye, what you will find that as compared to the say normal or the second say abnormality myopia, here you will find that there occurs reduction in the anterior posterior axis of the eye, and eyeball say size increases in the say it's a vertical corrected by convex lens. Say these are the same say that is the abnormalities. That is say just give a say look at this point, you come across two terms, far point of my far point and the near point of the vision. Near point is the that is the say nearest distance where I perceive the image very clearly, and the far point is the that is the limit of the eye where say it can see the object which are placed at say a far distance approximately 6 meters. And the far point of myopia is say that is more as compared to the near point. Similarly, here say it shows this say that is the myopic eye. What is the peculiarity is that say since there is an increase in the anterior posterior axis of the eye, the image which is formed is in front of that is the retina, since the light rays are converged in front of retina. And how it is corrected? It is corrected by that is the say that is the concave lens, which here helps to concentrate or focus this say or converge this say light rays on the retinal surface. In the hypermetropia, since there is a say that is reduction in anterior posterior axis of the eyeball, and the image is not formed say on the retina, but it is formed say behind that is the retinal surface. And how this abnormality is corrected? It is corrected by that is the say convex lens, and that is the correction is by that is a convex lens, which here helps here to cause say that is the convergence of light rays on that is the retinal surface. Refractive errors affect the visual acuity. We have seen that visual acuity is nothing but the ability of the eye to perceive details of the object, or it is the ability to perceive any object clearly by the eye. Now how this say refractive errors are say that is the identify. There is a simple technique by which we can identify these refractive errors, that is by using certain test types, or we call test charts. These charts are called as a Snellen chart. Now what you are observing before you is the Snellen chart, which indicates say that arrangement of the different rows with a different size letter. The first letter is say capital E, and then the smallest letter say it shows here say starting from P, E, Z and so on. Now this chart is placed at 6 meters, and the individual has to read this say all letters clearly from that is a distance of 6 meters, means approximately 20 feet. If he is able to read say this letter at 20 meters, means visual acuity is 6 by 6, and this person is having say that is the normal vision. Similarly, say this chart is used just to test the visual acuity for that is the far placed object, or that is for far sightedness. Now there are different types of these say charts. For illiterate individuals, we can use say this type of the chart, where instead of letters we can use that is the different say that is the animal sizes, and they are used to read from the same distance of 6 meters, and say from that we can determine that is the visual acuity for far vision. Now this type is called as the say the C type of chart, a Landau chart, and the principle is same. The individual has to read this chart from that is the 6 meter, and you have to identify the opening of this, say that is the C ring in, or opening of this ring in the specific direction. Similarly, there is the other chart what you are observing here before you. It's called the J chart or Jagger chart. In Jagger charts, you will find different, say, sizes of the letter. These are called as, say, the letters are, say, identical to the printer's font type, say, letters. Nowadays we call it as a points, letter size as a point in the terms of our, say, the computer technology. Now what you will find that the letters are arranged from, that is the ascending order, from smaller size to the larger size. If the person can read, that is the first line, say that is the 0.37 m, it means that the person is having, that is a good near vision. This chart has to be read at a distance of, that is the 30 to 35 centimeter. By using this chart, we can determine, that is, say, that this refractive errors for, that is the far vision and that is the near vision. Now coming to the last abnormality, what we call it as the astigmatism. Now we have seen that astigmatism occurs due to the that is inequality of the corneal surface and that of, that is the lens. The parallel rays do not come to a point of focus on the retina. Unequal power of the refraction in the eye in more than one meridian. Blurring of vision occurs, headache, eye strain, watering, and ocular fatigue occurs. In severe cases, monoocular diplopia or double vision is observed. Now it is mainly caused due to the error in curvature of, that is the cornea, alteration in the curvature of the lens, or error in the curvature of, that is the eyeball. And how it is corrected? It is corrected by, that is the cylindrical type of the lens. Cylindrical type of the lens. The last aspect what we are going to study is the electroretinogram or ERG. Electroretinogram testing is an important diagnostic tool for patients with, say, retinitis pigmentosa. The ERG measures the function of rod and cone photoreceptors. Electroretinography measures the electrical responses of various cell types in the retina, including the photoreceptors, rods and the cones, inner retinal cells, the bipolar and amacrine cells, and the ganglion cells. Electroretinography, clinically used mainly by ophthalmologists and optometrists. The electroretinogram is used for the diagnosis of various retinal diseases. The first type is inherited, that is a retinal degeneration in which ERG can be useful. It includes the retinitis pigmentosa, that is nothing but degeneration of rods and cones, and related hereditary degenerations. Disorders mimicking retinitis pigmentosa. Second type is other ocular disorders in which the standard ERG provides useful information, which include here diabetic retinopathy, retinal detachment, assessment of retinal function after trauma, especially in vitreous hemorrhage, dense cataracts, and other conditions where the fundus cannot be visualized. How this ERG is recorded? ERG is recorded in comfortable position, either lying down or sitting up of the patient. Dilation of the patient's eye with standard dilating eye drops is done, and anesthetic drops are also used. In open eyelids, contact lens electrode or an electrode resembling a fine thread on each eye is placed. Indifferent electrode is also used, which is placed on the forehead skin. During an ERG recording session, different visual patterns such as flickering lights, dark adapted or scotopic, reading light adapted or photopic readings are observed. Retinal cells emit small electrical signals when stimulated by certain types of light. The ERG machine records the resulting electric signals, amplitude, that is what we call as a voltage, and time course. Now this is the basic waveform which is observed. You can see commonly the two components that are most often measured are A and B waves. You can see in this figure, this notch A and the peak is B. A wave is the first large negative component, followed by B wave, which is corneal positive and usually larger than A, which you can see. This is what we call it as a biphasic waveform of ARG of a normal, say, that is the patient. What is the meaning of different components which is recorded in this ERG? First is the A wave, which indicates initial corneal negative deflection derived from the cones and rods of the outer photoreceptor layers. B wave indicates here corneal positive deflection derived from the inner retina, predominantly Muller and say on bipolar cells. C wave is derived from the retinal pigment, epithelium, and photoreceptors, and D wave indicates that is the say non-functioning of the bipolar cells, or what we call it the off bipolar cells in other terms. Okay, this completes here. This ends here. That is the second part of, that is the human vision. And I just thank you for watching, and just I hereby request you to please that is subscribe my that is channel, so I can say improve that is my further series of lecture. Thank you very much.